So today we're talking about the roles of torsional stress and topoisomerases in DNA supercoiling. Torsional stress is a type of stress characterized by twisting while one end of the object is held in place. So when a double helix of DNA is put under too much torsional stress, overwinding occurs, resulting in supercoils. Today we'll be demonstrating supercoiling on a pair of headphones. So allow this headphone cord to represent a double helix of DNA. As we twist one end of the cord, applying more and more torsional stress to this end, and the other end is held in place, eventually the cord coils back over on itself, resulting in a supercoil. So this supercoiling process actually occurs naturally as DNA is unwinding. As DNA unwinds, more and more torsional stress is applied to one end, and the only way that the DNA coil is able to release tension is by coiling back over itself and forming supercoils like this. So as the strand of DNA continues unwinding and more tension builds up, the only way for it to keep unwinding is for some of the tension to be released periodically. One way to release this tension is to cut and splice a piece of the strand, unwind it, and then resplice it together. This is the role of topoisomerases. We have learned about two types of topoisomerases. These topoisomerases are enzymes, again, that move along the strand of DNA, cutting it, unwinding it, and resplicing it together in order to relieve tension. Topoisomerase 1 does this by cutting just a single strand of DNA, bringing it around the supercoil, and then reconnecting it to relieve the tension. While topoisomerase 2 does this by cutting both strands of DNA and unwinding the entire thing, relieving tension from up to 2 supercoils at once. But both of these enzymes make their way along the entire DNA strand, cutting and splicing and unwinding, relieving tension until the entire thing is completely relaxed and back to its normal shape.